Yeah. Hi, hello everyone. So it's good to be the first speaker of the session. Nobody would fall asleep, I hope. So yeah, today I'm going to talk about the famous dipterocarps that constitute nearly 80% of all the emergent trees that we find in Southeast Asia. So before starting off, I would like us to recall the major plate tectonic movements with respect to India. Around 180 million years ago, India was a part of a huge landmass called Gondwana land, and it was situated in the southern hemisphere. Now, with time, all the continents of the Gondwana landmass started to drift apart, and this Indian plate also started to move from southern hemisphere to northern hemisphere. It entered into the tropics around 70 million years ago, and then straddled at the equator around 56 million years ago before finally colliding with the Southeast Asia around late Eocene, around 40 million years ago, and then it attained its final position. Now, when India was straddling at the equator, the global temperature conditions were quite high, both at the poles as well as at the equator. So here we can say that pole equator temperature gradient was quite low. But as time passed, the temperature started to decline and this pole equator temperature gradient became very prominent. Now, this temperature pattern or the variation in the temperature pattern is also reflected in the distribution of tropical rainforest or tropical biodiversity. So when the temperature was high, the tropical-like biome or the tropical biodiversity was expanded up to the higher latitudes. But as we move towards geologically recent time, these tropical-like biomes got restricted to the low latitudes. Now, these tropical-like biomes or the tropical rainforest cradle 50% of the global plant species. So we were interested in tracing the origin and migratory history of tropical rainforest with respect to the plate tectonics and changes in climatic conditions in deep time. We were also interested in investigating the underlying causes for the retraction of the biodiversity or the tropical biodiversity from the higher latitudes to lower latitudes. So to achieve these objectives, we decided to study the origin and paleobiogeography of the plant family called Dipterocarpaceae, which represents the Southeast Asian tropical rainforest that in turn constitute nearly 15% of the worldwide tropical rainforest. So before moving on to the dipterocarps, I would like to briefly mention about this conflict that we generally see in the molecular and fossil ages of any taxonomic group. So here, this molecular phylogenetic tree suggests that angiosperms evolved around 200 million years ago in Triassic. But the fossil records of angiosperms have been reported from 130 million years old sediments. So this particular study on dipterocarps would also put forward a methodology that, if not resolved, but definitely suppresses this conflict. And how do we do that? We'll get to know in a bit. Okay, so Dipterocarpaceae is a pantropical megathermal angiosperm family that is subdivided into 3 subfamilies. Pacaramoideae, which is endemic to South America, then Monotoideae, which is restricted to South Africa and Madagascar. Then the most diverse subfamily Dipterocarpoideae, which is distributed in South Asia and Southeast Asia, and one of the species is also distributed in Seychelles. Now, to look out for Dipterocarpaceae fossils, we collected sediment samples from 4 Indian geological sites. So here, site 1 is in central India, that is around 68 million years old, and the rest of the 3 sites are lignite mines that are situated on the western India, and they are around 63 to 54 million years old. Additionally, we also processed some samples from the Northeast Africa, and these samples dated back to 68 million years old. We recovered 8 Dipterocarpaceae type pollen fossils belonging to genera Dipterocarpus, Dryabalanops, Shoria, Parashoria, Bateriopsis, and Monotus. Now, the extant relatives of all the fossils within this red rectangle are currently distributed in South Asia, so it is acceptable to have their records in Indian sediments. But we also recovered fossils of Bateriopsis and Monotus that are currently restricted to Seychelles and Africa, respectively. These are the Dipterocarpus type pollen fossils. This is from central India, around 68 million years old. This is from Western India, around 55 million years old. And these 2 are from Africa, again 68 million years old. Then we have Diabalonops type pollen fossils, then Vatica, Monotus, Vateriopsis, Paraschoria, Shoria albida, Shoria robusta. So next, to evaluate the phylogenetic placement of these fossils, we constructed a morphological matrix that included the pollen morphology of all the available extant as well as fossil Dipterocarpaceae species. And then we analyzed this matrix using these two softwares that gave these two topologies as an output. So these two morphological phylogenetic topologies help us tease apart the position of our fossils within the different clades of Dipterocarpaceae phylogenetic tree. Next, we downloaded the DNA sequences of all the available Dipterocarpaceae members in GeneBank and then analyzed it using these two softwares to have the evolutionary relationships among different Dipterocarpaceae members. Now, to avoid the conflict between the fossil and molecular ages, we integrated the morphological dataset with the molecular dataset and ran the combined dataset in a software called Beast2 that provides us the divergence times for the different lineages of Dipterocarpaceae. Now, this dated tree is then analyzed or processed using BioGeoBS and R to reconstruct the ancestral areas of all the lineages. Now, this is a lot to absorb. So what I did, I just combined or summarized all the trees that I just displayed into a simplified version. So this is the simplified version of Dipterocarpaceae evolutionary phylogenetic tree. Here we divided it into 5 different clades, and each clade represents distinct genus. And each internal node represents the ancestral ages or ancestral areas for the genus subtended by it. For example, if we look here at Dipterocarpus, the stem lineage of Dipterocarpus evolved around 90 million years ago in Africa, and it dispersed to India around 70 million years ago. Now we wanted to compare these reconstructed ancestral ages and areas with the paleoclimatic and geodynamic conditions in the past. So we constructed these paleoclimatic maps for the time interval that were quite relevant in the Dipterocarpaceae evolutionary history. Here, this light green color represents the seasonal climatic setting. This dark green color represents the perhumid, extremely wet climate, and this yellow represents the aridity. So the tree suggests the family originated around 100 million years ago. But if we look at the paleogeographic or paleoclimatic maps, India was situated too south in the southern hemisphere during this time to support any growth of any megathermal angiosperms, let alone Dipterocarpaceae. And it is also evident by the lack of angiosperm fossils in the Indian sediments during that time. However, this seasonal climate was prevailing on the northern parts of Africa, which could have supported the growth of Dipterocarpaceae. Now, unless stated otherwise, it is usually assumed that the fossils in the past were thriving in the climatic conditions equivalent to the modern day setting in which their living counterparts are flourishing today. So, based on this concept, the study suggests the stem lineages of Dipterocarpus, Shoria, and Monotus evolved in the seasonal climatic setting of Africa, whereas the stem lineages of Diabalonops and Vateriopsis radiated in the perhumid climatic conditions. Now, by Maastrichtian, all of the crown lineages had already evolved, and around Maastrichtian Paleocene time, these genera dispersed from Africa to India via a chain of islands called Kohistan-Ladakh Island Arc. Now, the important point here is this Kohistan-Ladakh Island Arc itself was ecologically segregated into perhumid as well as seasonal regimes, and that allowed the dispersal of Dipterocarpaceae genera of differential climatic preferences. Now, around Paleocene-Eocene time, an outburst in the lineages of Vatica and Shoria is observed under the perhumid climatic setting of the Indian plate. And this outburst indicates that the family underwent an ecological release during that time. Or in simpler words, we would say that the family radiated so rapidly, maybe because of the availability of new niches or maybe because of the conducive climatic conditions. Then, as India migrated further towards the north, it got aligned with the Southeast Asia within the same climatic zone. I should have shown here the Southeast Asia, but never mind. I mean, during this time, India and Southeast Asia were climatically aligned that facilitated the dispersal of Dipterocarpaceae members from India to Southeast Asia. And when India finally collided with Southeast Asia, aridity transgressed over the Indian subcontinent, due to which the perhumid lineages of Bateriopsis got wiped off, and the wet lineages of Dipterocarpus, Monotus, Shoria, and Vatica, they got restricted to the perhumid regions of southern Western Ghats. Next, this lineage through time curve of Dipterocarpaceae suggests that the family underwent high diversification around 20 million years ago in Southeast Asia, giving rise to the magnificent Southeast Asian Dipterocarp lowland tropical rainforest. So to summarize it, I would just say the Southeast Asia lowland Dipterocarp rainforest originated in the seasonally wet climatic conditions of tropical Africa around 100 million years ago. Then it got dispersed to India via KLIA around 70 to 65 million years ago, and then finally spread to Southeast Asia following out of India dispersal hypothesis. Now here, the major driving force for the evolution and diversification of Dipterocarpaceae was the ecological segregation of KLIA regions into perhumid and seasonal climate that provided favorable climatic circumstances for the easy dispersal of Dipterocarpaceae. And we should also note here that the dispersals event could only take place when two land masses were geologically aligned within the same climatic zone. So this is all what I have to say today. Thanks.
Excellent. Do we have questions? So if you have a question, either raise your hand and we'll call on you, or type them in the chat or in the question and answer feature. Looks like we have a question from Neil.
Okay, so great talk, by the way. But one thing I noticed looking at your BioGeoBears results was there seemed to be loads of transitions from Africa to India, but only a couple that I could see from Africa to the Americas. Do you have any explanation for that?
For Pachyramoideae, so we were interested in the evolutionary history of Dipterocarpoideae here, and Monotoideae as well, but we did not include Pachyramoideae because genetic sequences was not available for them. So that was the limitation we had.
Yeah. Okay, fair enough.
Yeah.
Thanks.
Other questions? Oh, there's one in the chat that says, just wanted to get your thoughts that currently we do not have many Dipterocarpaceae in the WG as compared to SL and SEA.
As compared to Sri Lanka and Southeast Asia, yeah, that's because of the climatic conditions, of course. I mean, there is the climatic conditions that are favorable for the Dipterocarpaceae growth are present in patches in southern India, and it is spread across vast regions in Sri Lanka and Southeast Asia. So that is one of the most probable explanation that can be given for this.
Excellent. What other plant families could be forming tropical Asian forests before the arrival of the Dipterocarps?
Myristicaceae or Ebinaceae. I mean, I have also worked on Ebinaceae. The evolution of Ebinaceae is also contemporaneous to this Dipterocarpaceae. Then, Myristicaceae also we have recovered from the same assemblage. Then Anonaceae. We have recovered plenty of families actually within the same fossil assemblage from which we have recovered the Dipterocarps.
Excellent. Excellent. We should probably move on to the next talk now. Thank you.
Thanks.
And our next speaker is Jarnavi Joshi. Jarnavi, you put your slides up. We'll wait just up, and it's 9:15. So begin. Thank you.
Hello. Thanks, Mahi, for the excellent talk and introduction to Peninsular India and Gondwana biogeography. It's very helpful. I'm Jannavi Joshi. I'm assistant professor at Center for Cellular and Molecular Biology in Hyderabad, India. And today I'll be talking about diversification dynamics in Peninsular India of tropical Asia And this work is led by a graduate student, Pragyadeep, in the lab, and he's also in the audience today. So one of the most striking pattern and consistent pattern is that we have highest diversity in the tropics. And researchers have been very fascinated to understand these fundamental questions about their origins and maintenance such high diversity. But specifically to look at and identify, characterize the processes which are responsible for generating differential species diversity even within the tropical regions, as well as distinct clades. And diversification analysis allows us to do that. And we are particularly interested in the peninsular India of tropical Asia. As Mahi introduced us very beautifully, that peninsular India was part of Gondwana land, and then it sequentially broke away from Antarctica, Australia, Africa, collided with Asia. And as a result of this complex geographical past, what we have today are many species which are Gondwanan origin and then radiated in peninsular India, species which disperse into India from Southeast Asia and have endemic radiations, as well as from Eurasia. Apart from being a part of Gondwana landmass, peninsular India also had a massive Deccan volcanic activity around 66 million years ago. And this activity affected northern and the central part much more as compared to the southern parts. As Mahi really beautifully explained and showed us those maps where how climate also kept changing as peninsular India was moving during this period before it collided with Asia, and climate also kept changing as it changed its position across the latitudes. We also had periods of intense aridification and monsoon intensification, and as recently as glacial maxima. And it's been believed that this geoclimatic past has influenced the species diversity which we see today. We also have contemporary seasonal gradients as one moves from south to north. Here is a Western Ghats, which is a global biodiversity hotspot along with Sri Lanka. You have seasonality gradient where southern parts are less seasonal as one moves towards north is less seasonal. We also have another mountain chain, Eastern Ghats on the eastern slopes of east coast of West Peninsular India, where climatic gradients are inverse. Northern areas receive much more rainfall as compared to the south. So both past geoclimatic history as well as the contemporary gradients have given rise to varied habitat types. We have tropical wet forest, we have montane shola forest, dry forest, and also these lateritic plateaus which are stunted with wet evergreen forest in between as a result of Deccan Trap volcanism. And the biodiversity here is quite unique. Many lineages are endemic. What has changed in the last 2 decades is that many new species discoveries are happening across groups. Here is a graph for annulars and lizards, and many of these new species discoveries are also using molecular tools. And which means we don't only have a distribution data now, we also have time calibrated species trees for these lineages. And when we started looking at these data sets, we realized we have around 42 lineages or time calibrated trees for at least minimum 4 species, and which are endemic to peninsular India. And they vary in their diversification times. As you can see, the soil arthropods, which are the oldest lineages here, which are around 150, to the bird lineage, which is a very young lineage. Unfortunately, we don't have many bird and mammal endemic lineages in peninsular India. Largely, the endemic diversity is comprised of herpetofauna and arthropods, and many plant groups which are yet to be, I guess, studied. So when we started putting together these data sets, we realized we have approximately at least 700 species, of which 630 are endemic, and we have a molecular data. We have a distribution data for all of them. We know their crown age, and we know the species richness in each of the lineage. They also occupy very distinct habitats within the peninsular India. Some are restricted to the wet forest, some are in both wet and dry habitats, some are freshwater species. And here is a graph where you have on the y-axis number of lineages and how are they distributed to these different habitat types. The second graph shows you this is the taxonomic identity of these distinct lineages and biogeographic affinities. You can see soil arthropod and mollusk are mostly Gondwanan. Amphibian reptiles have both Asian and Gondwanan lineages, versus birds and plants are predominantly Asian lineages. And as Mahi just told us that what we are calling plants as Asian because Currently, the diversity which we have are of Asian origin. You may have had Gondwanan elements which are being wiped out because of the Deccan Trap volcanism, and today's diversity is largely of Asian origin. And given this background, we were curious to know what are the diversification rates and how have they varied through time in peninsular India, and what are their biotic and abiotic drivers of these patterns. So first we looked at mode of this diversification, and based on the literature and intrinsic factors of the organism, literature suggests that plants to have higher diversification rates as compared to the animals. Given we have both Gondwanan and Asian elements in the peninsular India, Gondwanan elements in peninsular India are largely wet zone specialists, suggesting they are retaining their ancestral niches, showing niche conservatism. And if it's true, they will have lower diversification rate as compared to the young Asian taxa, which have much wider niches. And secondary, if you remain in the wet forest and you haven't diversified in the other forest, you will have a lower diversification rate, versus lineages which are occupying both wet and dry may have higher rates of diversification. So to test these predictions, we fitted constant birth death models to all 34 lineages. What you have here on the x-axis are the different taxonomic groups, and on the y-axis, speciation, extinction, and net diversification rates. You can see largely speciation rates are driving the diversification rate. Across taxonomic groups, extinction rates have remained low. Secondly, soil arthropods have the lowest diversification rate, and the plants had the highest diversification rates as we had predicted. Again, Gondwanan lineages, which are gray in color, have a lower diversification rates, versus Southeast Asian lineages had higher diversification rates. Interestingly, we didn't find a pattern between the wet and dry forest. However, freshwater lineages had a lower diversification rate. However, our number is only 4, so we can't very say it conclusively. But wet and dry forest lineages didn't differ in their diversification rates. Then we looked at what is the mode of diversification. As we know, in tropical forests, people have hypothesized that diversification rate can remain constant, being the oldest and more stable biome. If that's the true case, then you will have a gradual accumulation of diversity. Alternately, lineages can also show non-uniform diversification, where you can have an early increase in the diversification, which is followed by saturation. And this is also attributed to the ecological limit or niches which limit the diversification. And in context of peninsular India, once the wet forest niches are occupied, you can have a saturation. It can also be because of bouts. You can have a second scenario where there are bouts of increased diversity, but then again a loss of diversity, waxing and waning, because of multiple geoclimatic events which were very drastic in peninsular India. Or you can have abundant of niches because constantly the climate was changing, and if species have actually used those changing climates and diversified, showing something like adaptive radiation. And to test these scenarios, we again fitted constant birth death models, but also time varying birth death models using both R Panda and Comet. And what we found is that most of the lineages showing gradual accumulation, and few lineages showed both saturated and waxing and waning. However, we know that lineages can also have episodic rate shifts. So we performed a Comet analysis for that. But Pragyadeep also wrote a small function to detect local episodic rate shifts within a lineage. And we fitted both Comet as well as his function to all these 34 lineages. Then, interestingly, our numbers of gradual accumulation decreased, but still 19 lineages still show a constant accumulation, few of them with episodic rate shift. Given this, we went on to ask now, what are the biotic and abiotic drivers of these lineages which have diversified in peninsular India with these distinct patterns of either constant diversification or with varying time. So first we looked at explicitly ecological limit hypothesis through diversity dependent models. And among the abiotic variables, we looked at temperature dependence, as Mahi showed us one of the graphs where the temperature had changed quite dramatically throughout the last 60 million years, which is what most of the diversification for peninsular Indian groups have happened. We used onset of seasonality or Himalayan origin as one of the proxy for this. We also looked at intensification of aridification, which is thought to have influenced the diversification in peninsular India. And when we looked at these drivers with respect to this lineage diversification, what we found that around 9 lineages show the temperature dependence, and 3 lineages were ecological limits or diversity dependence. But it wasn't alone. It's always with a temperature dependence. And as you can see, there wasn't one particular scenario for a taxa. All Gondwana lineages show gradual accumulation, except one where you had saturated accumulation and a redshift. This light blue is for mollusks. All of them show gradual accumulation. In amphibians and reptiles, it was a mix, both time varying as well as constant diversification. Birds show constant accumulation, and plants also had both time varying as well as constant diversification. Interesting part is all the rate shifts are in the Miocene, mid-Miocene. And this is one of the geological event which has thought to influence peninsular Indian diversity. And we also show that many of these rate shifts through comet as well as through Pakideep's function, which detects these episodic rate shifts, are in this time period.
3 minutes.
Just to give you this flavor, like how many lineages which had gradual accumulations, we found 19, 2 lineages which were saturated, and we found the drivers was temperature dependence as well as density dependent. Both saturated and accumulation was found in 1 lineage, waxing and waning in 4, and symmetrical redshifts were in 9. However, we didn't find any signature of adaptive radiation among these 34 lineages. We further looked at these lineages in ordination framework to see what are the drivers of these lineage diversification. On the x-axis here are the loadings of both diversity and diversification patterns, and on the y-axis is predominantly the characteristics of lineage identity. Just to simplify this graph further, you can see the scenario which is constant diversification is forming one cluster which is significantly different from temperature or density dependent. And within this temperature and diversity dependent, they form very distinct clusters. Also, if you think about taxonomic identities, soil arthropods and mollusks are very well separated from amphibian, reptiles, birds, and plants, which form one cluster. But interestingly, Gondwana and Asian elements as well are separated largely on the dimension 2, which is the lineage identity, as we see earlier as well. So just to put these results in a global perspective across tropical regions, what do we know? So we do see that historical stability has been important, as 19 lineages show gradual species accumulation through time. Another hypothesis which has been put forward for tropical diversity is that higher rates of speciation. We do see across 15 lineages where paleotemperature, Miocene aridification, ecological limits have played an important role. Another implication of these results is thinking about if the tropics have been thought to be an old age biome. So the older lineages, do they dictate the species richness and gives rise to the high diversity? So we tested for both clade age and diversification rate hypotheses. And what we find is that richness is better predicted by mid diversification rates than the species richness. And it's interesting to see because this has not one group of organisms, but across soil arthropods, reptiles, amphibians, and plants, we get consistently a pattern that species richness is better predicted by diversification rates. And that's it. I'm happy to take questions.
Excellent. Thank you. I don't think we have any time for questions, but if you have questions for Janavi, you can put them in the chat and she can respond to them there. Our next speaker is Mylan Vitek. Do you want to put your slides up, Mylan? And thanks. It's good to start.
Yes, thank you. I will just start my presentation. Okay, so welcome everyone. I'm Milan, and I'm a researcher at the Czech Academy of Sciences in Czech Republic. And I would like to talk to you today about our projects on parental senescence effects in annual killifish. To start, I would just like to differentiate what I mean by aging and what I mean by senescence. So to me, age is just a number. Senescence then is the physiological decline that is related to age, that is happening in physiological and reproductive functions. And it varies between individuals. It has a genetic basis, and partially is affected also by the environment. And despite the aging, the chronological age is objective. The senescence is happening on different rate between individuals within population. This individual variation and the genetic basis of it is a substrate for natural selection. And the main hypothesis about how senescence evolves is based on this hypothesis of selection shadow, that after the individual starts to reproduce, from that moment on, the selection declines. I mean, the selection on the phenotype, because once the genes are passed to the next generation, any maintenance of the body towards later ages is kind of like not really necessary. Also, the decline is not uniform across different traits in the body, as is this prediction by Morat and Newsey, who state that wherever there is a senescence in fecundity, the senescence in maternal effects will be quicker. We study senescence in this model species. It's an African annual fish, turquoise killifish, that is characteristic by its adaptation to these ephemeral ponds in the African savanna that last during a short period of rainy season. It extremely unfortunate years or regions, the individuals may not even be able to reproduce, so they really have to live fast. And by a natural selection, the duration of the pools, their lifespan is condensed. On the other hand, they survive the dry season in the stage of embryos. That can take several months. Sometimes it takes multiple years. And after reproduction, they do not care about their offspring. Our aim in this project was to compare the trajectory of aging in the number and the quality of the offspring. We did it in the lab with the long-term monitoring of the individual fish and their status. We collected data on their body size, like length and weight, and we also collected data on their reproduction, the number of eggs they spawned, fertilization rate, and egg size. Regarding the offspring traits, we selected clutches at specific moments of the parental fish life. So the first moment corresponds to the maturity of the fish, when the survival of the population is 100%. The second one is a moment when half of the females were still alive, and then when only 25% of the female is alive. We performed this specific crossing scheme within a family. So we bred 3 males and 3 females together within a family, and collected the clutches from these combinations to incubate them for 6 months. One of the offspring traits that we assayed was their survival of this extended period that corresponds to the duration of the dry season in the natural habitat. And the second one was their early growth rate. So we measured how fast the fish grew during their first 4 weeks of life after they hatched. So we have the preliminary results, and so we focus on the senescence of the parents, and then how does the chronological age of parents affect the offspring viability. So you can see here that males and females kept growing until quite late. Females continued until basically the end of the experiment. This is like first trait where we looked at the decline of their condition. Simplified, it's just index, basically body mass index, like the proportion of weight to body length. The individuals, the dots above the zero, have better condition than average, and below zero, it shows that they are in poor condition. So you can see that both in males and females, there is a trend to decline with increasing age. The reproductive traits, you can see that in all three traits that we monitored, there is a trend to decline with age, although because of the females grew throughout their life, they initially increased their fecundity, and towards the end of life, however, you can see that the number of eggs is going down. With the proportion of fertilized eggs, it's steadily declining, and with egg size, there is kind of plateau, but towards the end, there's also apparent decline. To kind of generalize this data, we attempted to put these three measures together, but it's topical only for the females, because like males have little effect on the number and egg size in these fish. And with this plot, I would like to illustrate that you can see that the females that have mean reproductive performance low always happen to be in the lower part of this chart compared to the females that are in the upper half, meaning they perform better consistently. It's not completely random. So it's showing that we have good quality individuals and low quality individuals during the whole experiment. Now to the parental effects, the offspring survival, we saw quite clear difference between the survival of offspring from the young parents, as you can see here, compared to the two groups that were collected from parents that were older. Now to the results on the early life growth. We expected that the trend will be similar as in the survival, like the survival will decline with the parental age. But as you can see here, both in the females and males, the trend is not really there. There is no decline related to age. So this did not happen. To summarize it, I showed you that there is a clear age-related decline in parents across multiple traits, mainly related to female reproductive traits. The parental senescence also affects the survival of the offspring, but not the early life growth. So this can mean that once the embryos survive the dry period, they are basically at the same level, and it can be caused by condition-dependent embryo survival. Simply just the weaker embryos do not survive the dry period. And just to give you a bit of outlook because our project is kind of still continuing. We want to complement this data with study of cellular markers that were collected throughout the study. So we are looking at the DNA methylation that is the topic of the presentation of my colleague Malahat that is going to talk to you in approximately half an hour. And we will also look at the changes in the telomere length. And this is done in collaboration with the local lab. So sorry, I still cannot present the data now. I would like to acknowledge the people that participated various ways in this project. Also the funding body. I would like to thank to the organizers of this meeting and the technical help, and I would like to thank you for your attention. And if you have any question, I'm happy to try to answer, or you can email me, or up to you.
Excellent, thank you. So we do have time for questions. Anyone? I have a question. What do you think is driving the lower survival of the initial embryos? Do the embryos during the dry period, are they at all metabolically active? So it could be a provisioning problem?
This is a good question. You know, like it's kind of annual organism in many aspects. So they have indeed stage of dormancy. Actually, they have 3 stages of diapause. They are really morphologically conserved. So they have 3 moments where they can enter resting stage, where the consumption of the energy is really, really, really low. But still, the metabolism is not stopped. So we measured the exercise. I do not present the results on it. So we may connect the exercise as a kind of like proxy of the energy invested by the female into the eggs. But now I cannot give you exact answer.
Right, because I know there's some insects that diapause over the winter. They need, some amount of energy to get through the winter. And I was just thinking maybe this is the same thing for the killifish.
Sure, but at least with the populations that we keep in captivity, we saw that some of the embryos, at least, they are able to survive like 3-4 years in the lab. So
wow,
not sure whether this happens also in the field, but it's really, really difficult to find the eggs. Our lab is doing some research on this, but not really sure how topical this multiple year survival is for the field conditions.
Great. Any other questions?
I guess I could jump in with a question.
Sure.
Do you think that multiple generations of this treatment would lead to perhaps like stacking epigenetic effects on the embryos? So maybe you'll find some sort of signature if you repeat this multiple generations later.
Yeah, this is really good question. It would be possible, but the experiment to achieve this would be really challenging because having multiple strains and the capacity of the fish breeding system. But yeah, it would be really interesting. I think what we saw from our results, maybe the offspring that is really affected by the senescence is dying along the way somewhere. So maybe there will be no parental effect, but it's really difficult to say.
Great, thank you.
Thank you.
We probably don't have time, but Mylan, there's one question for you in the Q&A feature. You could respond to them there.
Yeah, okay. Thank you very much.
We should probably move on to the next talk.
Sure. Thank you.
And our next talk is by Arleti Roy. Arleti, do you want to put your slides up?
Yes.
And begin whenever you're ready.
Okay. Hello, everyone. My name is Arlette Roy. I'm a PhD student working at the CNRS in French Guiana. And today I'm going to talk about the transparency of a group of chemically defended butterflies called Etomines. So in nature, organisms use a variety of defense strategies against predators. So at first glance, two categories seem to stand out. On the one hand, those who seek to hide from predators, represented by cryptic strategies like background matching. And on the other hand, those who seek to be seen and recognized by predators with the very well-known example of aposematism, which is the association of a toxicity and a honest warning signal, like a particular color pattern. But aposematism and cryptism are now often seen as two extremes of a continuum. And so why are they intermediate strategies or signals that are not maximized over this continuum? So to answer this question, we chose to work on the butterflies of the Etomini tribe. This is a highly diverse group of butterflies with approximately 400 species. And as you can see, most of them display a bright color pattern, and they all are considered chemically defended. Yet, 80% of Etomines are also partially transparent in their wings, some more than others. And so why have these butterflies evolved to be transparent, and what are the potential advantages toward predators? So since these butterflies are present in different microhabitats, we hypothesized that this may be in response to different type of predators, be they avian or arthropod predators. And we also hypothesized that detectability may differ with distance from the observer. So first, we attempted to ascertain if transparency decreases detectability for these different predator types. So we took more than 300 photographs of 15 species of Etomines, plus two cryptic non-toxic controls, one opaque and one with transparent wings. And we use a model called quantitative color pattern analysis with the ImageG software, and with several predator vision models, so the Blue-tit and the Salticidae. And this model is based on three criteria: the abundance of the cone of the eye of the chosen visual system, its visual acuity, and especially the distance of the prey from the observer. So at close range, we found that transparency decreases detectability for birds and not for arthropods. And for both birds and arthropods, we found that transparency decreased the distance at which they were no longer detectable, making the more opaque species still detectable at greater distances than the transparent species. So for the Blue-tits, transparency decreased detectability irrespective of distance. And for the Salticidae, transparency decreases the detectability not at close range, but at a distance. So as I said, we also tested some control species that were considered cryptic. So for the opaque species, the detectability and the distance threshold was always inferior to all Etomini butterflies, for both the bird and the spider. And for the transparent control, the detectability and distance threshold was on average equal to the transparent Etomines for the Blue-tit vision, but was inferior to all Itominis for the Salticidae division. So now that we know that transparent Itominis are less detectable for birds and arthropods, we ask ourselves, are they any less toxic? So thanks to a previous study by Bellamy McClure in 2019, we know that for birds, transparent species of Itominis are not any less unpalatable. And in fact, the speed of avoidance learning by chicks was similar for transparent and opaque Itominis. But if transparent Itominis are less toxic for another type of predator, it could explain the need for a more cryptic defense strategy. So to determine if transparent species are less well protected against arthropod predators, we tested those same 11 species of Itomini with plus one non-cryptic and non-toxic butterfly called Cisia proba, using wild Nephilia spiders. And we tested each spiders only once with one Itomini and one Cisia. So in general, Itomini butterflies showed high survival rates compared to the palatable butterflies, which were always killed and eaten. And moreover, transparent Itominis showed higher survival rates than opaque ones. However, transparency did not correlate with ingestion. And in fact, all Itominis were rarely eaten, if at all. So we know that thanks to previous studies and with what we observe in the field, that in average, opaque and transparent Itominis don't fly at the same heights. Opaque species fly usually high, whereas transparent Itominis fly pretty close to the ground. So these differences in microhabitat could make Itominis encounter different types of predators. And so the transparent Itominis could therefore encounter more ground arthropod predators, for example. And we know that birds are capable of recognizing and avoid aposematic signal, but arthropods lack the long-term learning capabilities to really integrate this type of defense strategies. So transparency could then allow butterflies to evade detection at long distances while deterring predators with their toxicity if they actually get caught. And so this offers a significant evolutionary advantage in diverse ecological settings. Thank you for your attention.
Excellent. We have plenty of time for questions. Just raise your hand if you'd like to ask a question, or type it in the chat if you don't feel like speaking. Nope. So Arleti, do you think that, which do you think came first, the transparency or the toxicity in this group?
Well, so the transparency is actually a derived trait. So if we look at the phylogeny, the more basal species are opaque and toxic, along with the cystoclade donainae that are also opaque and toxic and with bright color patterns. So, so it's the transparency that came after.
So if transparency works mainly against birds, and the toxicity is where the birds actually learn to not eat them.
Yes.
Why do you think they would both come up at the same time against? Why would you need transparency if the birds already learn not to eat you?
Well, the birds can learn for both opaque and transparent species that they are not good to eat. But I think for me the importance of transparency lies on the fact that there are other predators than birds. that cannot learn this type of mechanism and cannot retain the information that this type of pattern and coloration will actually mean that this is not good to eat. So for me, the transparency came for protecting from those predators that cannot learn.
And there's a question in the chat. Are they opaque / transparent, or is it a continuum from transparency to opacity?
Yeah. So actually, there is some completely opaque species. Maybe I can show. Wait. Sorry. So wait. Yes, there is completely opaque species, and then well, when we have a lot of species, 400 species, and I think maybe like 2,000 subspecies, and so in this huge amount of species, there is all type of transparency levels and like coverage on the wing.
Mm-hmm. And there's a question in the Q&A that asks, you mentioned the difference in microhabitat between transparent and opaque butterflies. What lighting conditions did the analysis correspond to?
For my photographs?
Yeah.
So we took the photograph in a control settings. We took in a light box, so with always the same lights. But obviously in nature, if you are closer to the canopy, you will have more light. But for the pictures, we wanted to have a control setting and to compare the detectability for each species if they were at the same height in the forest.
Yeah. We still have a couple of minutes if someone else has a question.
I can pop in.
Sure.
Is there ever hybridization that occurs between these two species and that lead to much reduced fitness in the hybrids?
There is hybridization, but only between two like subspecies, and usually they are pretty close in their color pattern. So I don't think it would make a real difference with hybrids.
Okay, so they're quite set between their transparency and
and within species, there is little differences in morphological patterns. They're really like all the same.
Got it.
And one more question in the Q&A that asks, are there any predators that have adapted to eat these butterflies or might be seeking them out specifically for reduced competition? That can deal with the toxicity.
Actually, I don't know. For me, not that I know of. I know that I have a friend who talked me about the fact that he did some experiments with other insects like mantids, and they also rejected the butterflies. For birds, actually, I don't know. I don't think so.
Excellent, thank you.
Well, there is some. There is some species of Atomines that seems to be less unpalatable. Like in the paper of Melanie McClure, there is a species called Serratinia tunia that is that seems to be less toxic for birds.
Good.
But
I think we need.
In general, I don't think so.
Excellent. I think we'll have to move on now. Great talk. Thank you.
Thank you very much.
Malahat, do you want to put your slides up?
Yes.
You can begin when you're ready.
Yes. Can you see my presentation?
Yes.
Okay, hello everyone. I'm Malahat Dionat. I'm a postdoc researcher in Institute of Vertebrate Biology, Czech Academy of Science, Czech Republic. And I will talk about DNA methylation as a marker or mediator and transmission to the parents by aging. First of all, we need to know what is senescence. I know that my colleague Milan explained, but it's good to know that gradual decline in an organism condition with age called senescence, that it's an ongoing, inherent, and a complex process. The onset and rate of senescence can be different between individuals and function. And when the parents aging, their care on their offspring diminishes, it means that age of a parent affects the quality and survival of their offspring. We are still at the beginning of understanding the effect of parental aging on their offspring, but to know this, we need to understand the correlation between the level of senescence in parents and the characteristic of their offspring. Also, we need to find the physiological changes by aging happens in the parents and if they transmit and inherit to their offspring. To do this, I am using a marker, methylation as a marker, epigenetic marker, and methylation is adding methyl to the cytosine when the cytosine are next to the guanine, and we call it CPG sites in the genome. Actually, if methylation happens in the promoter of the genes, this can change the gene expression, and we are using this epigenetic marker because they mainly affected by changes in the conditions by environment or stress, pollution, or aging. And this is a good marker to study how these changes transmit from parents to the offspring. To study methylation, I am using Notobranchius fuseri, Turco's killifish that Milan explained about it, because they grow very fast and they have very short lifespan, and no parental care reported for this species. The aim of this study is to know the amount of DNA methylation in the somatic tissue in the fin of the parents by aging, and to know whether these changes or this changing in the amount of DNA methylation inherit to the offspring or not. To study methylation, I am using next generation, one of the methods of next generation sequencing that it called EpiRat sequencing, is a modification of double digest restriction site associated DNA sequencing. In DDRat sequencing, actually, we just use one pairs of enzyme, EcoR1 and MSP, but in our method EpiRat sequencing, we are using two pairs of enzyme. It means that we prepare library for each sample 2 times, once using the EcoR1 and MSP, and once with EcoR1 and HPA, because MSP is insensitive to methylation. It means that it cuts the DNA and doesn't care about the methylated site in the DNA, but HPA is sensitive to methylation. It means that it doesn't cut the DNA if it is methylated. Then the sequences that come from these two libraries, we compare them together to find the methylation pattern and detect the amount of methylation. The study design is using the parental generation. We are using 33 samples, 6 male and 5 females across aging when they are in age 1, in age 2, and age 3. And actually, we are testing the 55 samples of the offspring coming from these different ages. After having the sequences in hand, we check the quality of the samples, and then to do assembly, we use the reference genome of Notobranchius fusariae, and we extract the read count. Then we check them for normalization and filter the insufficient loci, and then change them to binary data. And finally, we do the differentially methylation analysis. First of all, I should mention that I didn't use all of the loci after assembly, and I wanted to work with the loci that are represented well across the samples. That's why I used the sequences coming from the library, which is not sensitive to methylation, the one that comes from MSP, to filter the absent loci. And I found that by increasing the minimum number of reads per locus across samples, the green one, the number of loci decrease. But when I was looking at the proportion of methylation, the red one, I saw that by increasing the minimum number of reads per locus, the proportion of methylation stabilized here somewhere in 33. That's why I use this number as a threshold, and the number of loci decreased to 3,485. I did another filtration to remove the loci that didn't show any differentiation, or they were not methylated across the samples, and the number of loci decreased to 926. Here I am showing the heat map. Here there are samples, and here I am showing the loci, and the gray parts, it's showing the level of methylation for samples across loci, and the white part is showing the loci that are not methylated. The dendrogram also grouped the samples that they show similar pattern of methylation. As I said, I wanted to know the amount of methylation first for parents. Here you can see the box plot for parents, female and male. And when I was checking this, I found that the amount of methylation shows relatively high individual variation. For example, here, if you look at the parents female, by aging, you can see that, for example, the amount of methylation for sample F1 is increasing, which has showing the different pattern with another sample. If I choose C7, you can see it shows a different pattern, and it was the same for male. By aging, the amount of methylation is dependent on the, it shows a lot of variation individually. And when I checked, compared the parents with the offspring through aging, I found that the amount of methylation increased by aging in the parents, but you can see here that I don't see this pattern for the offspring. It means that the amount of methylation by aging in parents doesn't mirror in the offspring. And to summarize my talk, I can say that the main thing that affect on the amount of DNA methylation is dependent mainly on the age of the individual, and it means that they don't inherit to the from parents to the offspring, and probably they reset in the zygote after fertilization. But what is the next step? As we have a very good annotated reference genome of Notobranchius fusariae, we are interested in the changes that happens by aging, to look at the loci that they change their status by aging in the parents. For example, when it goes from age 1 and age 2 to age 2, we want to look at the loci that become methylated, or vice versa. If there is any loci that from age 1 to age 2, they become unmethylated. And we want to see if the status of methylation in parents is the same as in offspring. And also we are interested in the functionality of these genes using the reference genome. Thank you very much for your attention. I don't know why I cannot show the next slide, but it's nothing. It's just thanks for your listening.
Thank you. Excellent. Other questions? Just raise your hand or type them in the chat. I had a question. So you did all of the studies of methylation on fin clips, correct?
Yes.
So what is the relationship between the degree of methylation in fin clips and in gametes that would be what would be being created for the DNA complement for the offspring?
Sorry, I couldn't understand the question. What is the difference between the fin clip and?
And what happens in a gamete? So a female making eggs, right? The
Yes.
offspring is going to only be susceptible to methylation in eggs and sperm.
Actually, we also collected the gonad samples and still we didn't do the analysis, but I can say that the amount of methylation in the gonads were much higher than the fin clips, but still we didn't do the analysis for the gonads, but it's in process. We will do it.
Thanks. Other questions? We still got a few minutes.
Have you considered looking at methylation in other tissues, such as maybe liver, like any other tissues that are metabolism related rather than fin clips?
We didn't do this. No, we just collected the tissue from fins and gonads.
Other questions? No. Well, we've still got a couple of minutes till our final talk in the session. Albeit, you're up. Good. So we still got 3 minutes, though. Let's wait until.
Okay, that's fine.
Do you want to try your slides out just to make sure they're working good?
Yeah. Yes.
I guess I have a question that just came to mind.
Sure.
You said you were going to be using the gene annotations to perhaps be looking at the differentially methylated loci. Do you have any predictions on what types of functions might be affected by methylation that changes between ages?
I'm sorry. Could you please repeat your question?
Oh yeah, no problem. You mentioned that you're going to be using the gene annotations to look at the functions of these differentially methylated loci, correct?
Yes, we want to look at the functionality of the loci that change their status by aging.
Yes, yeah. Do you have any predictions on some functions that you expect to see in those differentially methylated
not
regions?
yet. Exactly. I finished this analysis maybe 1 week ago, and maybe I am learning this how to find the I mean that for now, for example, I know like 10 genes that they change their status from age 1 to age 2. They were methylated. And now I am learning how I can find these genes on the reference genome, because I have the coordination of the genes, and still I am learning how to find their functionality.
Okay, great. Thank you. I'm excited to see what you find.
Excellent.
Okay, can you see my slides?
Yes, excellent.
Okay. Okay, so this is I'm Aviat Chefetz. I'm from the Open University of Israel. This is a game theoretical work trying to think about the origins or the reason behind sex bias dispersal in cooperatively breeding birds. And the motivation for this is empirical and experimental work I've been doing in the last several years with my colleague Roni Ostracher on the Arabian babbler in the southern part of Israel. So let me try to show you first maybe a short clip on the Arabian babblers, and then we can take it from there. So let's see.
Day breaks over the Shizaf Nature Reserve in the Arava Valley. Members of the babbler group stretch a wing and open an eye to the new morning. The group has spent the night in a cuddle and will keep in close contact and touch throughout the day. Morning wash, allopreening, playing, and foraging together. This is a typical day of the babblers, the only cooperatively nesting bird in Israel.
Okay. I hope you could see the slides. Let me. So can you see back my slides now? Can you see?
Yes.
Yes. Okay. So this was a short clip on the Arabian babblers, and the question is more general about the sex bias dispersal in cooperative breeders. So it is known that in most bird species, in order to breed, females disperse more than do males. But the case of cooperatively breeding birds is much more complicated. And that's because, as you saw in this example of the Arabian babblers, these groups are territorial, and they have what could be called an incest taboo. That is, I hatch in a particular group, or I manage to get out of the nest. I look around and I see this is my extended family. I'm not going to mate with any of them. So my only option to mate is either by dispersing into another group where I don't have direct relatives, or waiting for somebody of the other sex to join my group, and in such case, I would be able to breed with them. Now we have the classical explanation of Greenwood of why males disperse less in birds, and that's because they defend territories. But in the case of cooperatively breeding birds, this is much more complicated because, as in our study in Shizaf Nature Reserve in the Arava Valley, the entire area is saturated with territories of Arabian babblers. So it's not about forming a new group in a new territory. It's either it's one of the two options that I mentioned before. Could I somehow lost view of everybody? So could I get some indication if I can be heard well?
Yes.
Yeah, we can hear you.
Okay. Okay. So if we think about it, because dispersal is so costly, okay, I have to leave my known territory, my known group, my colleagues, so to speak, to the group, my group mates, whom I know, and enter a new territory, maybe pass through other territories in order to join another group. That's definitely dangerous. And we know that survival among dispersers is lower than those who stay at their natal group. Then there is an obvious kind of game theoretic interdependence, right? If females disperse more, then males have a higher incentive to wait for them and not to disperse, and vice versa. Okay. But if we look at the existing literature, theoretical literature about the sex bias dispersal, we don't have predictions, okay, that some say anything specific about females versus males, okay? So there are either theoretical models that say, you know, one sex would be completely philopatric and only the other one would disperse, or it could be that, but without saying whether these are the males or the females who are dispersing. Or there are also models where each sex disperses with some proclivity, but again, without saying which of them, okay? So we wanted to try to understand here what is the asymmetry between the sexes in cooperative breeders in birds that would establish such a sex bias in favor of the males and making females disperse more. Okay, so here are some, you know, stylized facts, okay, about dispersal in cooperatively breeding birds. So first of all, you know, there's no deadline for dispersal. That's what's called delayed dispersal, right? So nobody is kicked out of their group if they stay, okay? But of course, if you're already sexually mature, then okay, you would like to breed. Of course, dispersal can be informed, okay? So it's not a statistical or a stochastic thing, okay? You know, I can respond to information, to opportunities. You know, has somebody of my own sex disappeared from a neighboring group? Maybe that's an option for me to try to take their breeding vacancy. And at least in the Arabian babblers I know well, they really collect a lot of information. They do these border patrols, they shout, they fight, and this way they collect a lot of information. Generally, you know, I may like to disperse either for breeding directly with somebody in the target group, or it could be also sometimes with somebody from a neighboring group. It happens in the Seychelles warbler, for example. And importantly, if I'm a male and I disperse into another group, then I will not be welcomed by the males there, okay, because I'm a competition for breeding there. And likewise for females, they wouldn't be liked by the local females in the target group. And here's the asymmetry, okay? Males are heavier and stronger, typically. And therefore, they like females joining their group. They are more able to defend them and welcome them than females welcoming males, okay, and overcoming the opposition of the local males. Okay, so here's a simplified model that in which we try to look into this asymmetry and the effects of this asymmetry, okay? So in this very stylized model, okay, every morning, okay, non-breeding adults, okay, you know, they wake up, they look around, and they get signals about what happens if they disperse today. And they get some signal if they take their best shot today, what is the chance that they fail? fail means either I perish on the way to the target group, or maybe I reach the target group, but then I don't manage to join, okay? Or maybe somehow I manage to join, but then the females there don't want me, or whatever, okay? This is my failure probability. And because of the asymmetry that I just described, we assume that there is what's called first order stochastically domination in male dispersal failure distribution. That is, for every cutoff Z, the probability for males that they fail, the probability that this dispersal failure is larger than Z is larger for males than for females. Okay, that's the equation here. Okay, and what we like to look at is cutoff strategies. Okay, so I wake up. If finally my dispersal failure is smaller than this cutoff M, then I disperse. And if I'm a female, I may have a different cutoff. I disperse if I see that today my failure probability is smaller than F. Okay, and in this example, let's assume that for males, the failure probability is uniform in 0, 1, and for females, it's in a smaller interval between 0 and A for some A smaller than 1. Okay, so I disperse if X over A, so my probability of failure is smaller than this cutoff F. Okay, so here's the heart of the model. Okay, what's my expected fitness today is, okay, I either get a good signal, I disperse, and that's the probability that I succeed. This is the probability, average probability I fail, and this is the probability I succeed. Or I don't disperse, a female joins and I sire offspring with her. Or if I stay and a female doesn't join, then I survive the night with probability delta smaller than 1, and tomorrow is another day.
3 minutes.
Yes. Okay, so you see that this expression appears here and here. So I can isolate it. And by maximizing this expression, I find what I call what game theorists call the reaction function. Okay, if females use the cutoff F, then I better use M. Okay, I can do the analogous calculation for the females. Okay, and we can see that the intersection of the two reaction curves is what would be the ESS, the evolutionary stable strategy, that is actually also continuously stable. I will not get into this. And here is how this changes with time, as the degree of asymmetry increases. So smaller A, more asymmetry. Then we can see that the threshold for males go down, so they disperse. They take lower risks in dispersal, the more asymmetry. For females, somewhat surprisingly, they first are willing to take more risks, but then a little bit, and then a little bit again, more risk, but then a little bit less. Okay, so okay, let me jump forward. Okay, so let me just tell you how this is compatible with the empirical evidence. So the first finding is that indeed females disperse more than males. Okay, and this is a well-known phenomenon. But the important thing from this model is that females tolerate more risks at dispersal. Okay, and what's the evidence for that? Okay, so in most cooperatively breeding birds, they disperse to farther away target groups. Okay, so that's more dangerous. They do so at a younger age. And in particular, in the Arabian babblers, they also disperse in smaller coalitions. They disperse more frequently alone. And they somehow sometimes spend more time between leaving the group and between joining the new group. So again, a dangerous period. Okay,
Thank you.
thanks.
Excellent. We don't have any more time for questions. The session's now over, but if anybody wants to hang around and ask questions, we'll keep the Zoom going for a few minutes.
Okay. Thanks, Mark, for this. Thanks, Mark, for this very gentle chairing of this session in a remote and in this remote framework. And thanks everybody for the interesting talks.